I uploaded also a document in the content that described exactly what I want you to do. Okay, so please pay attention and follow up my instructions. Key point, I will not accept anything that is not in an MP3, an MP4 format. Okay, because last year I had many issues when students said just share that document from Google Docs or Google Slides, and then I needed to have authorization and I needed to see each slide separately, not in a continuum, and it's painful, particularly when I need to watch the many of you, okay? So MP4 submitted, I send you some formats into indications, some videos into how to do it, just in case you don't, because very, very, very simple. You can even use your Zoom and record it. It's just 5 minutes, and it recorded right straight into MP4 format, okay? And anything into Dropbox, okay? Every submission has to be in Dropbox, because if you send them into my mail, it can be buried among all the many emails I receive per day, okay? Okay, so now that I have said that, we are moving into chapter 6, is sex determination and gametogenesis will be the material of our first and second lab. So just let me
I have a question before we start.
Yes.
We still don't have topics for a research paper, right? Do we choose them ourselves, or are you giving a specific topic?
For what, Anish?
For the final research paper.
Oh, no, you haven't that. You don't have that. I will give that topic later. So you have a term paper that is due later on. I can't remember when I did say that will be, but that I have it here somewhere in my notebook. So the term paper, I'm gonna give you that in February, okay? Because it's due in April 2nd.
Okay, and you give those topics out. I was just wondering if you help us get us started.
I give the topics.
Okay.
Okay. So I am the one giving the topics because I'm the one marking. So I need to know the material before. If it's just something that I don't know, well, I need to learn at the same time that I'm marking. And it won't be fair for you guys. I need to know well the material. Okay, so don't worry about it. For the presentation, the video, the outline must be submitted by January 29th. So you need to start looking into the papers so you can get some ideas of what you want to be covering, okay, for the video.
Sounds good. Thank you.
Okie dokie, not a problem. Okay, so now I am going to share my screen with you guys, and we are ready to go. Where is my Zoom? Sure. Here it is. Okay. So okay, so in this chapter, we're going to be seeing things that through the semester, okay, where we see that we have used animals as a source of experimentation in order to better understand the different phenomena that relates to the creation, formation, and development of vertebrates mainly. As I say, we're going to be using the sea urchin as a model, but most of the information going to be related to vertebrates. So we will cover different stages from development of gamete, fertilization, and different developmental stages. And this is the first one, is the sex determination. Even though this happens later during development, it is important to know first what determines sex. Last week, we talked about how the environment in which cells are found is capable of playing a key role in what this type of cells become. And also, we know that the environment can play a key role in the energy storage, in nutrient, and can determine the amount of offspring that a female may have, right? If conditions are good, normally many females are capable of having a little more babies, but if conditions are not so good, maybe the babies won't be as much, okay? So in this chapter, what we will be seeing is that in many species, sex is also determined by environmental factors such as temperature. So when the field of physiological embryology was first developing, the obvious environmental variable to manipulate were temperature and the media in which embryos were developing. In mammals, with the specific chromosomes X and Y, when an individual have their second chromosome, their second chromosome of XX, for instance, we know it's a female, right? But if it's a XY, it is a male. In birds, it is the opposite, okay? The male has two similar sex chromosomes, that is the ZZ, and the female has the unmatched pair, that is ZW. In fish, the Y chromosome does not play any role at all in sex determination, but rather number of X chromosomes will determine the sexual phenotype of the individual, okay? And of course, we will discuss mammals and other vertebrates during this chapter. So not always we have this matching. Oh, that's not what I wanted. Oops. Here. So not always we have this pairing to determine one sex. We can have other things that can determine the sex of individuals. So in humans and in mice, the Y chromosome, we know that is critical in determining the sex of the individual, the offspring. So XX in mammals are usually females, have ovaries, and produce eggs. During meiosis, all eggs produced have an X chromosome. Now, the XY in mammals are usually males, as we mentioned prior, and have testes, make sperm. During meiosis, half of the sperm have an X chromosome, and the other half have a Y chromosome. So there are many more possible combinations that are the ones that we see here in the screen. And depending on the combination, we can obtain, as we see here, a 50-50, right? 50% females and 50% males, depending on how this combine. Now, nevertheless, this does not answer the main question about how sex is determined in mammals, because it's not just about how these chromosomes are matching. There are so many other things, and maybe today, and I don't know, maybe of you already know, maybe you don't know, but we're going to be seeing things that are a little different. And this is one of them. What? Even myself, after I had my PhD, when I started learning this, I'm like, what? My God, I didn't knew that, you know? So we always learn a little more wiser every night. So in mammals, sex is determined by gonad formation and by different sets of hormones produced by those gonads. So even if you have the right type of chromosome, hormones going to play a very important role in finally determining whether you will be a male or a female, okay? So there are two things. In mammals, we have two determination periods. We have the primary determination and we have the secondary determination. In the primary determination, we define these as the determination of the gonad, okay? Egg-forming ovaries or sperm-forming testes, okay? That is determined by chromosomes, as we have seen in the previous figure and we see in here. So this is the bipotential gonad. So all of us, whether you were a male or a female at the very beginning, we have bipotential gonads. So this is very early during development, okay? So these gonads are, say, indifferentiated, okay? So what it means that as these gonads continue growing, then they're gonna have two things. We have the Mullerian ducts, okay, here in orange, which normally we know belong to females, right? But also they're gonna have the Golgian ducts, okay? And this one here in green, we know that those are for males, okay? So if it's a XY, the gonad become testes, okay? And here we have the testes that start developing, and the Wolffian ducts persist. Now, if you have the XX chromosome, the gonads become ovaries, and the Mullerian ducts will persist. We are clear up to here? Okay, now, then once we have determined a little bit of this, whether there is a Mullerian duct or the Wolffian duct, the one that's going to persist, we're going to have the secondary determination. This secondary determination is a determination of the male's or female phenotype. So here we are not talking about the genotype, but the phenotype. And this is made by the hormones produced by the gonads, okay? So with this determination, hormones will cause the external genitalia to develop either in the male direction, we're going to see the development of a penis and an scrotum, or in the female direction, there will be a clitoris, labia majora, okay? So this is what we see here in this little table here. So we have the gonad. So gonad type is either testis or is an ovary. And then we have the germ cell location inside the testes or in the follicles, okay, near on the cortex. Which of the ducts persist? So if it's a male, is the Wolffian, and we're going to have the vasa deferens, epididymis, and seminal vesicles. This is what the Wolffian ducts become, okay? And then we have the Mullerian in the females, where we have the oviduct, uterus, cervix, and upper portion of the vagina. And then, of course, we're going to have the associated glands, the prostate, and the skinny glands. And then we have the scrotum or the labia majora, or the tubercle genitalia, that is the penis, or the clitoris, which are analogs. Now, when we talk about these factors that are going to be determining whether it becomes a male or a female, we know that the Y chromosome that determines the male phenotype depends on the time it takes for the sex determination factor to be triggered. So remember last week I told you time in development is of an essence. Here it is one of those examples, okay? So depending on the time that it takes these hormones or these factors to be triggered and to induce those cells, then they become a female or a male, okay? So at the beginning, we start as usual with a cascade of inductions to convert the genitalia, the genital ridge that we're gonna see in a couple of seconds in another image, into the bipotential gonad. That bipotential gonad is only created by the effect of these genes that have been triggered. Okay, so we have the SF1, WNT1, LHX9, the GATA4. Okay, those are the genes that will determine this ridge to become bipotential. Then, if any of these genes, just one, is missing, no gonad formation will occur. They all four are required for this genital ridge to become bipotential gonad. Okay. Now, to induce this bipotential gonad to form female ovaries, which is this line over here, we're gonna need to activate other set of genes. Where's my mouse? Here. Okay. So this set of genes are the FOXLY2, RSPO1, and WNT4. So once again, look at this. WNT, that is a wingless, and we say that we have like bazillion wingless, not bazillion, but we have at least 12. Okay. So here is the 4, and here is 1. Okay, so, and then we have these 3 genes need to be activated in this bipotential gonad to become ovaries. On the other hand, SRI gene on the chromosome Y is the testis determining factor, and SRI will in turn trigger SOX9. Okay, so in here we have an additional cascade because SRI is triggered first, and then SOX9 by SRI. But in case of the female, the 3 of them need to be activated almost at the same time. Okay, so different type of genes activation, and the time at which these are activated will determine whether it will become an ovary or whether it will become a testis. Okay, so for instance, if the gene SRI is present, it will inhibit the formation of ovaries, right? But it will induce the formation of testes. Remember, this is a bipotential gonad. And at this time, we have, as we saw here, both the Mullerian and the Wolffian ducts. Okay? Is this clear? Okay. Okay, so now that the primary determination has been directed, there is now the time for the secondary sex determination. And as I said before, it depends on the secretion of hormones, okay? And these hormones, of course, are secreted by each type of gonad. And of course, some paracrine factors. So this secondary determination generates a sexual phenotype outside of the gonads, okay? So female and male ducts and external genitalia. As we all know, for females, we are talking about estrogen is the hormone, right? So estrogen enables the production of the Mullerian ducts. We will then develop into the different parts. So the uterus, oviduct, cervix, and the upper vagina, okay? We have also this in here, we have the labia, clitoris, and the lower vagina. So just because females are a little more complicated, right? That's what males normally will say about us, that we are very complicated. We have more organs to create. So we have a little bit more of a tissue that need to be created. And on top of that, I forgot to mention also the mammary glands. The mammary glands are also part of the triggers of this estrogen stimulation. Well, for males, we are talking about only about 2 factors. We are talking about the anti-Mullerian hormone, the AMH, okay? And we are also talking about, what is it? I forgot it. So it's the the anti-Mullerian, that's what I wanted to say. The anti-Mullerian hormone is a member of the TGF-beta paracrine factor, okay? So we also know this as the Mullerian inhibitor factor, so MIF. And because it's anti-Mullerian, it means that it will destroy the Mullerian dogs, okay? And which means that you're gonna keep the Wolffian dogs. That is the one that is typical for males, okay? So the second factor is the steroid testosterone, okay? And testosterone, what causes is the masculinization of the embryo, okay? And will stimulate, of course, the formation of the penis and the scrotum, okay? The seminal vesicles and the prostate gland. So females is basically estrogen alone that is very potent, okay? And creates all the external parts, keeping up and stimulating the Mullerian duct. But for males, then we have, you see here, the SF1 factor gene that we saw before, right? This will activate the anti-Mullerian, which will cause a degradation of the Mullerian, keeping up the Wolffian duct, and the Wolffian duct integrity is maintained by the presence of testosterone. And then we're gonna have all the external structures, phenotype of male. Any questions regarding this? No? Okay, so now we're gonna go step by step about how this occurs. So in humans, the undifferentiated gonad, which means the bipotential gonad, appears very, very early in development. It's week 4. So we just barely know that we are pregnant, right? If we are like a clock getting our periods, maybe we are waiting for to have our period, or the oopsie, we are pregnant, right? And look at that, week 4, and we have already a gonad formation in this embryo, okay? So this gonad, this bipotential gonad, which have, this is the genital ridge in here that we were talking a couple of slides before, okay? It will remain indifferentiated up to week 7. So we are just creating the tissues, cells continue to migrate into this ridge over here, and we wait until week 7, more or less. So this ridge, as we see it here, expand the epithelium between week 4 to week 6. So we see it started there, but then it's continue expanding their epithelium, the germinal epithelium during those weeks, okay? The epithelium is associated mesenchyme. Remember, we saw the mesenchyme cells last week. So the epithelium and its associated mesenchyme continue to proliferate into the 2 ridges for the right and left gonad, okay? These genital ridges are somatic, not germ cells. So pay attention to that, because even though they're going to create the germ cells, these ridges are made of somatic cells, okay? The germ cells then start migrating into this gonad ridge in here at week 6. So this is why the week 6 is here is important. Those germ cells are migrated into this somatic ridge here, okay, in week 6. And we're going to continue with the development. By week 8, which is the one on top, we're going to see that the Sertoli cells start being differentiated. Okay, that only happens in week 8. The Sertoli cells, we know that play a very important role in the males. So these cells surround the incoming germ cells and form the testis cords, okay? So we see here how they start forming the cords of the testis, okay? Later, these cores will be organized into a network that is something that we call the rete testis. Okay, so the rete testis is normally the section where this sperm going to start going, migrating, and maintaining to mature. So these structures will later get encased with a thick extracellular matrix, which is called the tunica albuginea. So this already here in pink, okay? That's the tunica albuginea, and it will encase the rete testis. And the Sertoli cells will secrete the anti-Mullerian hormone, okay? And later they will support the development of a sperm. So we know that the Sertoli cells are the ones that stimulate the formation of a sperm, but at the very beginning, and all of this in week 8 to week 20, we see a way more development of those cords. The rete testis is going to come in this section, and the Sertoli cells would come and populate all these ducts, okay? Then we have the Leydig cells. The Leydig cells are the ones that make what the Leydig cells? Testosterone, right? So Leydig cells make the testosterone, and they develop from the interstitial mesenchyme of the differentiated testis. So we need these testes to be differentiated a little bit before the Leydig cells can be able to secrete testosterone. And testosterone, we know, is the secondary determination for the male phenotype, okay? Only at puberty, but after birth in other animals, the testis cores mature into seminiferous tubules, as we're going to see here. These are more of the seminiferous tubules, where germ cells will produce mature spermatozoa. Okay, so the efferent ducts are the mature remodel of the mesophrenic kidney. Don't worry, we're going to be talking about that later when we're going to be talking about the urinary system formation. But if I'm coming back to a couple of slides, where is it? Here, we see how the gonads are nearby the kidneys, okay? So they are forming all from the same part of the structure. And the epididymis and the vasa deferent will develop from the Wolffian duct. So those are the only two that want to create that. So Wolffian duct, the one that start in the bipotential gonad, are the ones that are going to finish being the epididymis and the vasa deferens. Any questions up to here for the males? Nope. Okay. Female. So I'm not going back into the week 4 because it's the same for male and females, right? They are bipotential gonads, and the same thing is the rich are formed from the somatic and the cells start migrating. Now, week 8 is the point of differentiation from that bipotential gonad. And so in here in females, the Mullerian duct remains intact. Okay, so we're going to see it right here. And then the oviduct, uterus, cervix, and the upper vagina are going to start forming from that Mullerian duct. Due to the low levels of testosterone in females, Wolffian ducts degenerates. Okay, so it's not because we have anti-Wolffian hormone like we have for males. It's just because the level of testosterone is too low in females. So as in males, the germ cells migrate near the outer surface of the gonad. So in here, and this is when we see the growth of cortical cords, of secondary sex cords, okay? And they want to mix with the gonal somatic cells. So we see that there is a little bit of a mix. It's not a continuous as it was in the males. It's a little bit more of a mixing. Near birth, each individual cell is surrounded by somatic cells. The germ cells become the ova, and the surrounding cortical epithelial cells become the granulosa. So we have here, this is our ovarian follicle, and we see that it's surrounded, right, by these layers. So the granulosa goes completely outside. So the mesenchymal cells then become the thecal cells, and both the thecal and the granulosa are what it forms what we call absolutely the follicle. This is anatomy 101, okay? And that follicle envelope of germ cells is what produce the steroid hormone, okay? So each follicle contains a single germ cell, which the technical name is the oogonium. We will enter meiosis at this time. So now, as I mentioned in the, I don't know, I think it was the fourth or the third slide, there are many factors that are involved in the determination of sexual phenotype. The genital ridge, which is bipotential, can develop testes and ovaries based on the expression of the genes, and the genes that we already saw, right? So WNT, WT1, WNT4, the LHX9, GATA4, and the FS1, okay? So in here, we have our two fates, the fates for becoming a male and the fate to become a female. If no Y chromosome is present, so this is the XX, these factors further expression of WNT4, this is what is really big over here, okay? And there is another hormone that is not, no, yeah, it is here, is the ERISPO1, okay? These are soluble proteins that bind to the cell membrane receptor and stimulates another one that we have seen, it was Friday, dishevel. Remember dishevel, the dishevel protein that belongs to the WNT pathway, okay? So these two factors, WNT4 and RESPO1, stimulate and attach to the membrane of the cell and stimulates the dishevel protein from the WNT pathway to produce copious amounts of beta-catenin. Okay, beta-catenin is, I call it one of the cat factors because you do whatever you want, and that cat, you can throw it to the air and falls back again into the forelegs. Okay, so beta-catenin is one of those molecules that we're going to see from now until the end. Okay, so these two force the dishevel protein from the WNT pathway to produce copious amounts of beta-catenin right here, which in turn create a positive feedback, okay? Because this beta-catenin then gonna produce more WNT4 and more ERISPO1, okay? So this is a positive feedback among this. This beta-catenin also gonna stimulate the ovarian pathway of development, okay? And third point in here, beta-catenin prevents, and this is why we have this double inhibition here. So beta-catenin prevents the accumulation of SOX9, that is typical molecule for the male phenotype determination, okay? So it's a double blockage here. So this is why beta-catenin is so important. Now, if the Y chromosome is present, that's what we have in here, the same factors in the genital ridge will activate the esri gene. Esri, remember, I told you that esri was the first one that was important, Okay, so S3 will be the first one to be activated. S3 is the one that binds to the enhancer of SOX9 that we have in here now as a pivot point for the male phenotype determination. Okay, and this elevates the expression of other genes, as we see later, okay, that determine the male pathway. Okay, S3 and SOX9 blocks beta-catenin. We know that beta-catenin is very potent, so these two also, this is where we have the double blockage from catenin to block SOX9, but from SOX9 also to block the effect of beta-catenin. So it is clear, this figure? Okay. Now, having the right genes doesn't necessarily mean that you will get the organ you expect. So work on mice has shown that the S3 gene of some strains of mice fail to produce testes when bred onto different genetic backgrounds. So this is possibly caused by a delay in S3 expression. So once again, time is of an essence in sex determination, okay? This could also be due to the failure of the protein to accumulate to the critical threshold level required to trigger SOX9. So if we come back, oops, where is it? We come back to this one. This one is so important, right? And this one. So this is why these two are the pivot point for these hormones. So if SOX9 is delayed because S3 couldn't necessarily be activated, then the male pathway will fail. Which means that by the time SOX9 is launched to be triggered, it is too late and gonad has been directed to become an ovary because, remember, SOX9 will inhibit beta-catenin, right? So, and this is what some of you guys have as a subject, hermaphroditism, okay? So we can have one type of gonad, but the phenotype expressed is different, right? So you have the gonads, but you don't have the phenotype that should go along with that phenotype. So hermaphrodites are individuals who have both ovaries and testicular tissues present in them, okay? So they have what we call ova-testis, a mix of both ovary and testis. Or also they can present one ovary and one testis, okay? Hermaphrodites can also be a result from the translocation of a Y chromosome to an X chromosome. That's another way how hermaphrodites can be created. And if the translocated Y is on the active X chromosome, the Y chromosome will be active and S3 gene will be transcribed. If the Y chromosome is translocated in the inactive X chromosome, the Y chromosome will also be inactive. So we have all of this showing specifically that SOX9 and beta-catenin, the time at which they are activated will finally determine what they will become. Okay, so here is an experimental delay of the 3 gene activation, only 6 hours, that led to the failure of the formation of the testes or the ovary. So here we have 13 hours, 15 hours, 18 hours. This is the laminin. So we see that it's present. Here we delay the expression of SOX, and then we see that completely degraded as the tissue. Okay. Now, excuse me. Despite of the primary formation of the gonads in either ovary or testis, this does not determine the full sexual phenotype. So what we see in this little figure in the side was the presence of the different ducts, Mullerian in orange and Wolffian in green. Okay, so in this original bipotential indifferentiated gonad, with those tubes, both systems are present, males and females, right? If the XY chromosome or gonads become the testes, the Wolffian duct persists, and if the XX gonad becomes ovary, the Mullerian ducts persist. That's what we say prior. So hormones are really the ultimate determination of the phenotype that the individual will have, either a penis and a scrotum or the clitoris labia majora. Okay, so the hormones are really the final, and that's what is the second sex determination factor. Okay, so this phenotype, as mentioned earlier, is given by the secretion of the two hormones, either testosterone or estrogen. This second wave of sex determination for mammals occurs during puberty. So we have already talked about the first part of the sex determination. The Mullerian duct, under the stimulation of the estrogen, become the uterus, cervix, oviduct, and upper vagina. Okay, but then we have that the mesenchyme in the urogenital swelling secretes inhibitors or WNT signals. Okay, so here we have the inhibitor of WNT. Okay, so we have 3 points to consider in this figure here. First, in the absence of WNT, the cuff, okay, is an antagonist, okay, signal. Estrogen modifies the genitalian tubercle into the clitoris, and the labioscrotal folds into the labia majora surrounding the vagina. Okay, so we're going to have a variation, so we can also have some inhibitors. 2, in males, androgens, either testosterone or dihydrotestosterone, okay, bind to the androgen receptor in the mesenchymal cells and prevent the synthesis of WNT inhibitors. So this is the inhibition that we see here. Okay, line here, and prevent the synthesis of that WNT. Thus permit WNT expression in the mesenchyme. Third, WNT is permitted, and it causes the genital tubercle to become the penis, okay, on the scrotum. So we can have an up-regulation or we can have a down-regulation. If we up-regulate, it becomes more of a female, but if we have a down-regulation, then becomes a male. So hormones, once again, play a very important role in determining the final fate or phenotype of the individual. Okay, so that was for mammals and humans. Okay, so while the sex of mammals is chromosomal and hormone determined, as we just saw, the sex of most snakes and lizards is determined by sex chromosomes at the time of fertilization. On the other hand, the sex of most turtles and all species of crocodilians is determined after fertilization, and this is determined by the embryonic environment. So genes not necessarily means that you're going to have what you think you're going to have, okay? The temperature of the eggs during a certain period of development is the deciding factor in determining sex. As such, small changes in temperature can cause dramatic changes in sex ratio. And this is one of the most known failed experiments for conservation of some species way back when. Now we know, so we know better, but we're still learning, especially with this climate change. If we think that things going to be normal, well, climate change will have an increase, which means that the species that have a temperature sex determination going to be dramatically affected. Okay, so often eggs incubated at low temperature produce one sex, whereas eggs incubated at high temperature produces the other sex that is not produced at low temperature, with only a small temperature window in which both sexes are produced. And this is what causes sometimes a little bit of trouble. So in this figure, we see that the abrupt temperature-induced change in sex ratio for the red-eared slider turtle, okay, where if the brood of eggs is incubated at a temperature of 28, so we have here the temperature of 28 in Celsius, and we have here the proportion male-female sex ratio, okay? So if the eggs are incubated at 28 degrees, right in here, the hatching will be males, because this is the proportion of males. So highly, 100% of the eggs will be males. If the brood of eggs is incubated at 31, just a couple of degrees higher, right? The hatching will be females. So if the brood eggs are incubated in intermediate temperatures, the hatching will be half males and half females. And that's what we have about in this section here. So between 20, 29, and 30, we're going to have a perfect ratio of males and females. And it's just a couple of degrees, right? Between 28 and 40 and 31, it's just a couple of degrees. There's a variation in the snapping turtle. I think I have, no, I don't have the little click. So in the snapping turtle, if the brood of eggs is incubated either cold, 22 degrees over here, or hot at 28 degrees, the hatchlings will be females. So female here and female here. And between these two extremes, normally males will be more predominant, which means just, once again, it's a tricky question, right? What is the right temperature to maintain those eggs to have the perfect proportion of males and females? In lizards, we have a little bit of variation. For instance, in the tree-line skink, sex is determined by sex chromosomes, where XY will be males, XX will be females. However, at low temperature, the environment component overrides the genetic sex determination, and all offsprings are males. So once again, having the right set of chromosomes do not determine necessarily what they will become, especially in this. So temperature can override the genetic component and create something else. In the European turtle, also, incubation at 30 degrees, females are predominant, but at temperature below 25, males are produced. And the temperature to have the perfect ratio is 28.5. So once again, different. In the alligator, here we have also the same. Low temperatures below 31. This is 31.5. We get females. Same when we go over 35. And anything in between will create 100% males. So once again, it's a very tricky. What is the temperature really to be able to have a 50-50 ratio? Temperature will decide. So we all need to think about this in global warming. And even though people say, oh, no, this is great, right? Well, there are many species that are so susceptible to this that we're going to skew the sexes, and then potentially this species will disappear. So the incubation temperature at which an equal ratio of males and females is produced is what we call the pivotal temperature or the threshold temperature for sex reproduction. So prior to recognition of this threshold temperature in turtles, conservation biologists routinely employ high incubation temperatures for sea turtles, for instance, in captivity. Okay, but then we're going to only have one sex, and that was a total failure. Okay, what happens with higher temperature? Incubation rate is reduced, right? Because it's higher metabolism, so embryos will hatch earlier, but then the sex were skewed. So the result was production of all females, and this was recorded in many different papers, and this is one of them, the Bergeron et al., that talks about it. It was a recent proposal also to use this temperature threshold as a conservation tool to produce more females of endangered turtle species. And this has been criticized because of the potential negative impact on population structure and mating systems. We know, of course, yes, females are the ones that are producing the eggs. The more we have of females, the better it is. But also males, if they don't have competition, male competition is very important to maintain the good set of genes, the good set of males, right? So it is very criticized, or at least it was in the late 90s, early 2000s. So some scientists now have suggested that global climate has change has the potential to eliminate the production of male turtle offsprings. If the mean temperature increased by 4 degrees, that is what it has been suggested. Okay, we think that by the end of 50 more years, temperature will be dramatically increased, even by 1 degree. And 1 degree for us, it doesn't seem too much, but for individuals that depend on the environment temperature to regulate, is very, very, very important. So we need to use these turtles to be good environmental sentinels and monitor these biotic impacts of predicted global climate change. Another point that is really important, and we maybe not pay too much attention to it, in this course we will, though, is the PCBs. PCBs are the polychlorinated bisphenol, and a widespread environmental pollutant that causes adverse health effects in many individuals. And we have found a lot of evidence that shows that PCBs are capable of disrupting reproductive and endocrine function during development. And this Bergeron et al. Paper that I just put down here showed that PCBs reverse gonad sex in turtle species with the ones that present this TSD, threshold temperature for sex determination, indicating that now they not only will be affected by the temperature, but also by the presence of PCBs in the environment. So they are a little disadvantaged, okay? Okay, continue with this pollutants and the effects on gonad formation. The enzyme aromatase, which converts testosterone into estrogen, very important hormone, can be affected by environmental factors. Interestingly, the primary sex determination in reptiles and birds is influenced by hormones, opposite of what we saw a little bit in mammals. So in mammals, primary sex determination, we say that it was chromosomal, right? Either XX or Y. Y. But in reptiles and birds, hormones are important in the primary determination of sex, okay? In reptiles, estrogen can override temperature determination, inducing ovarian differentiation even and masculinizing temperatures. So temperature, okay, as far as the hormones are determined for these reptiles, they don't care. So the sensitive time for the effects of estrogen and their inhibitors coincides with the time when sex determination usually occurs. So once again, time is really important. At what time cells are affected, whether they have already committed or they haven't committed to a fate. Okay? So the aromatase activity of the turtle MS is very low at the male promoting temperature of 25 degrees. At the female promoting temperature of 30 degrees, the ones that we just saw in a couple of slides, in 2 slides prior, okay? The female at 30 degrees, aromatase activity increased dramatically during the critical period of sex determination. And this is what we see here in this figure. So many widespread pollutants can have a dramatic effect, and this is what we see in here. We have the polychlorinated bisphenol compound, the PCB, that can act as an estrogen signaling. Okay, so PCBs can reverse the sex of turtles raised as males, and this is what we see. So we have this masculinization of frogs by low amounts of atrazine. Okay, so here in A, we have a very beautiful testis of a frog in their natural side, only having just a slight amount. It has 0.5 ppb parts per billion, okay, of atrazine. So not too much of an effect, okay? In B, we have the testes of the frog, both of them, right and left, in a natural site containing 0.8 parts per billion. So it's only 0.3 parts per billion higher than in this one. And look at that, the effect. Okay, so we can observe a severe testicular dysgenesis. It's not as big. They are like a third of the size, if not a fourth of the size, right? Because this is only one. These are two. Okay, so we have a dysgenesis of this gonad just with 0.3 part per billion more. And here in C, what we have is the testosterone in nanograms per milliliter, and we have the control. So we have control males, but we have high levels of testosterone, right? And in here we have males that were treated with atrazine, and this treatment, these figures here, the production of testosterone is only about a month and a half after they have been exposed once. Okay? So in here with the atrazine males treated, and this was treated with very 25 parts per billion. Okay? So we see that these males are incapable of producing normal quantities of testosterone, and even they produce less than female controls. Okay, so atrazine can heavily disrupt the formation of gonads, which then disrupt the formation of efficient gonads. And testes transform these testes in ovaries. Okay, remember, if we don't have testosterone, then estrogen will be prevalent because the levels are even lower than what we found in females. So we have many fish also and frogs that can be demasculinized, okay, just because of the presence of different substances in the environment. So this atrazine induces aromatase, and aromatase converts testosterone in estrogen. So that's why we have this demasculinization. Gametogenesis. So gametogenesis is the formation of gametes, egg or sperm. Okay. The formation of these gametes also start at the level of the primordial germ cell. Okay. this is when we have the PGC, primordial germ cells. Okay. This one here are bipotential too, and their fate will be determined by where they reside, whether they are placed in ovaries or whether they are placed in testicles. The decision, I'm putting the decision in between, how we say that, quotation marks. My English and my French just start getting mixed all the time, sorry. So the decision of whether they are become eggs or sperm depends on a coordination of factors produced by the developing gonad, as we just saw. Okay, so the primordial germ cells originate, and pay attention to this, in the posterior part of the embryo, and then they migrate to the gonads. So they are not born, in quotation marks, at the gonad tissue. They are far away, and then they migrate into the gonad tissue that are forming. And this happened, as we saw at the beginning, about in week 8 to 20. Okay? So there exists a set of proteins that seems to be involved in gamete formation across different animals. So we have those in here. We have VASA, okay, that is required for activation of germ cells specific messages. That is VASA, so the creation of specific messages. Then we have nanos. And nanos, they bind to a pumilio, another factor, and blocks the RNA translation, preventing the cells to be part of any germ layer and arrest the cell cycle. So nanos is very important just to arrest that cell cycle that normally is activated in any other cell of the body, because we know that these cells can only be activated when maturity arrives, right? Like in puberty. Then we have Tudor. Tudor is, their presence seems to cause a sterility in Drosophila. We don't know too much, or at least I am not aware about in mammals what it does necessarily. But too much can cause a sterility in Drosophila. And then we have a peewee. A peewee, together with Tudor, are involved in transcriptional silencing portion of the genome. So the primordial germ cells, excuse me, migrate into the bipotential gonads. They multiply as they migrate. So here we have where they are born, and then these are migrating and then going into the reach of the gonads. During this migration, these cells are surrounded by the cell secreting stem cell factors that we call that the SCF, secreting stem cell factors, and allow their migration and survival. If they are not continually stimulated, they may become in a different portion. They need to continue their migration. Okay, so as we observe in this figure, cells migrate through the gut. This is the gut section over here. So they come over here, they migrate through the gut, and they go dorsally. So this is the back. They go dorsally, and then you're going to start populating the ridge of the gonad. So in order to follow a pathway is highly coordinated by signals, the signals that we were talking last week, okay? And this will allow the gonad to create the difference between spermatogenesis and oogenesis. One of the most striking differences between the two processes is indicated in the cell meiosis. So female oogenesis, male spermatogenesis. Meiosis is initiated once in a finite population of cells. We know that females normally have the oocytes that they're going to have for the rest of their life, right? When in males, meiosis is initiated continually by mitotically dividing the stem cells. So as soon as puberty arrives, they're going to continue producing new cells every time. For us, the females, we're stuck. So wherever we do, wherever we pollute our oocytes, that's it. Okay. So in females, meiosis is initiated in the embryonic gonad, but in males only until puberty. And the rest, that's basic meiosis, things that you should know. 1 gamete produced per meiosis, but 4 in males. Completion of meiosis delays for months or years, well, only if the egg is fertilized, and meiosis complete on a daily or week basis. It all depends how much is used. For females, meiosis is arrested at first meiotic prophase and reinitiated only in a small population of cells, and meiosis and differentiation process continues without the cell arrest. Differentiation of gamete occurs while diploid in first meiotic prophase. Then in here is while haploid after meiosis ends. And all chromosomes exhibit equivalent transcription and recombination, and sex chromosomes exclude from recombination and transcription during first meiotic phase. So this is just basic biology. Now, in this figure, we're going to have another component, another molecule, morphogen transcription factor. It plays both roles. Retinoic acid. So retinoic acid with WNT and Sonny Hedgehog are going to be the molecules that I'm going to be mentioning the most through this course. So we have here the female germ layers, cells, and here we have the mesonephros. Okay, the mesonephros is part of the kidney structure. Okay, so in females, retinoic acid, short for RA, and WNT4 secreted by the mesonephros reaches the gonad and triggers meiotic initiation via the induction of the estradiol 8 that you see here. Okay, so this retinoic acid is a gradient and it starts in the mesonephros, goes into the somatic, but in the germ-line cells will stimulate the activation of estradiol 8. This is a transcription factor in female germ cells. Okay. However, if inactivated, and here we see the inactivation by nanus 2, these nanus genes can inactivate that estradiol 8, then the female germ are suppressed and it will follow mainly the male fate. So even if we have already established pathway, if something comes and disrupts the formation and activation of this estradiol 8, the cells won't be able to continue the female, but will continue the male fate. In males, as we saw, estradiol 8 is downregulated by FGF9. Okay. That's the first. The FGF is the fibroblast growth factor 9. So retinoic acid, same as we saw in females, is produced by the mesonephros, is secreted by that mesonephros, and is degraded by this molecule that we have here, the CYP26B1. Okay, it's an enzyme produced by the testes, as we see in this image. So that's all we have in here. This is before day 13.5. This is after day 13.5. And this blockage of this pathway over here then prevent male germ cells from initiating meiosis until embryonic day 13.5. So we see here is blocking. We can have meiosis before day 13.5, but after day 13.5, that blockage is not necessarily, it's activated by nanos2, which in the females was repressed, remember? And here then we have the male phase. So once male reaches puberty, retinoic acid is synthesized in the Sertoli cells and then induces extra 8 in the sperm stem cells, and meiosis can occur, but only at puberty in males. So all of this just to say that retinoic acid plays a big role in males and at females, and then they're going to have other factors that are going to inhibit or not inhibit the process, whether it's a female or it's a male, but extra 8 is a key point in both of them. Is that clear? Okay. Okay. So you should all have seen this in genetics. Okay. The meiosis. So sexual production, evolution, variation, and the transmission of traits from one generation to the next depends on meiosis. Okay. As you know, meiosis has 2 divisions without an intervening period of DNA replication, and homologous chromosomes pair together and recombine the genetic material. Okay, so in this figure, we have that summary of. So we have the leptoteny, okay, where is when the condensation of the chromosome at this stage. Normally, chromosomes consist of 2 parallel chromatids, green and blue and red in here. Then we have the zygoteny. And the zygoteny, the homologous chromosomes pair side by side, so they make a little bit of synapses in here, and we can identify them as the tetra or bivalent chromatids because they are paired, right? Then we have the pachyteny. The chromatids can be individually identified, and the crossing over may occur into this section. So we have here how they are pairing all the way. Okay, so we're going to have the crossing over, and the genetic material is exchanged at this point. And then we have the diplotin. And the diplotin, normally the 2 homologous chromosomes start to separate. We see like a little Velcro, right? It starts separating a little bit. And we have the chiasmata, various parts that we see in here and we have in here. Well, the crossing over is still occurring. And during this stage, there exists a high rate of gene transcription into these chromosomes. Following the steps are the metaphase, and it starts with diakinesis. Okay, and when the chromosomes are the more condensed, because we see them very clear as a chromosomes, and with the nuclear envelope start breaking up a little bit, so we can have the next steps. So in anaphase 1, segregation of the homologous chromosomes, they are pulled to the poles, and then in metaphase 2, we have the restructuring of the membrane, okay? And then we have anaphase 2, and all of these processes is what we call the interkinesis or interphase 2. So we have a period of rest that the cells and some species enter during meiosis, and no DNA replication will occur during this phase. This is a basic figure of the spermatogenesis that you know. You want to see this, we're going to ask you to do review this figure again in the lab for gametogenesis. Okay. So spermatogenesis consists of 3 different phases. The first one is the proliferative phase for the formation of the sperm. So the spermatogonia. So we have here the stem cells that want to have one will be transformed in a spermatogonium type A and B. One of them will remain as a stem cell, while the other one will transform, divide, continue. So then we have the meiotic phase with the 2 divisions to create the haploid cells. And then we have the third step is the post-meiotic phase that we call the spermiogenesis, no spermatogenesis, but spermiogenesis, that each individual cell is separated. So during this phase, we have all joined by the cytoplasmatic bridge, but then during spermiogenesis, they get individual sperm release, okay? So review your notes in the lab once we put them up, because I haven't done that yet, and be ready for that lab. For the females, during oogenesis, they rely heavily on hormones, paracrine factors, and tissue anatomy. During embryo development, between the 2nd and the 7th gestational month for humans, thousands of cells develop. They will reach about 7 million oogonia. Okay, this is what we see in this figure. So that's the number of germ cells, millions, 7, just before birth. You see? Only a handful of these oogonias will survive and become primary oocytes. Okay, so we see at birth, this number has dramatically dropped. Okay, the primary oocyte will remain on this stage for 12 to 40 years. That's this stage of diplotene that we talked a couple of minutes ago. So you see that by 10, there is a lot of girls that get the first period at 10. I found that so young, but a girl can be pregnant at 10. Okay, I was way more close to this because I was I'm a spoiled girl. I had my first period around 18 because I used to do a lot of gymnastics. I was part of the National League in Colombia, so I was training 7 days a week and about 7 hours a day. So it was a lot of energy, not to produce hormones, to produce a maturity of the organ, but to develop and maintain muscle. That was it was there. Okay. And then when we start thinking about maybe your age, maybe thinking about babies around here, look at that. We are so poor in our organ. We make pity, right? So this primary oocyte going to stay in there until normally after puberty, when all of this start cycle by cycle, they're going to have a whole whack of them that start going to mature under the effect of the LH, the luteinizing hormone. Some of those will resume the cycle of meiosis. Normally 5 to 6 oocytes going to start maturing, but normally one only will reach maturity and become Greffian. The rest going to just dissolve. Okay. And that primary oocyte will complete the first meiotic cycle and proceed to the second. And the increase of LH induce that oocyte maturity. So this is what we're going to become here. And the idea is where at this point, when it completes this first phase, the first polar body is out. Okay. So at telophase, one of the daughter cells contains hardly almost no cytoplasm, and that's the first polar body, okay? While the second contains a great amount of cytoplasm and becomes the secondary oocyte. So this one becomes the secondary oocyte, okay? We will discuss this later in the next chapter, what happened during fertilization, okay? So only in here ovulation and fertilize, and then it will resume, okay? I have just a couple of slides, and I think I had a question. Someone had a question. I'm just going to try to see. Okay. So it is 24. We want to finish these 2 slides on Thursday, okay? And we continue into the next lecture, okay? See you next Thursday.
Thank you, Dr. Marie.
Okay, Anish, if anybody has any questions, you can ask any question, but okay.
I actually have a question about Peruso.
Yes.
I'm not able to see